When you look into a mirror, you see a relatively symmetrical organism, one whose left half is essentially a mirror image of the right half. Surgeons, on the other hand, see a strikingly asymmetric organism when they open a person's thoracic or abdominal cavity. The stomach, heart and spleen, for example, are shifted to the left side of the body, whereas the liver is largely on the right side. On occasion, a physician will see a patient in which the left-right asymmetry of the visceral organs is reversed, a condition called situs inversus. Situs inversus is seen in persons with Kartagener syndrome, which is also characterized by recurrent sinus and respiratory infections and infertility in males. The first clues to the underlying cause of this disorder came in the 1970s when it was discovered that the immotile sperm from these individuals had an abnormal axonymal structure. Depending on the patient, the axonemes may be missing outer or inner dynean arms, central microtubules or radial spoke structures. Subsequent studies have shown that mutations in a number of genes, including those encoding dynean, heavy and intermediate chains, can cause this syndrome. It is understandable that such patients would suffer from respiratory infections, which depend on the clearance of debris and bacteria by respiratory tract cilia, and from male infertility, but why would roughly half of them exhibit a reversed left-right asymmetry? The basic body plan of a mammal is laid down during gastrulation in association with a structure called the embryonic node. Each cell that makes up the node has a single cilium. These cilia have unusual properties. They are missing the two central microtubules and exhibit an unusual rotary motion. If the motility of these cilia is impaired, as happens in mice harboring mutations in a flagella dynean gene, roughly half of the animals develop reversed asymmetry, suggesting that left-right asymmetry in these mutants is determined by chance. The rotation of the nodal cilia moves the surrounding fluid to the left side of the embryonic midline, as was demonstrated by tracking the movement of microscopic fluorescent beads. It was proposed that the extracellular fluid moved by the nodal cilia contains morphogenetic substances that become concentrated on the left side of the embryo, leading to the eventual formation of different organs on different sides of the midline. This proposal is strongly supported by experimental studies in which mouse embryos were raised in miniaturized chambers in which an artificial flow of fluid across the node could be imposed. When embryos were subjected to a flow of fluid in a direction opposite to that occurring during normal development, the embryos developed with reversed left-right asymmetry. Many cells of the body have a single non-motile primary cilium that is lacking both central microtubules and dynean arms. These cilia were ignored by researchers for years, but more recent studies suggest that they have important functions as antennae that sense the chemical and mechanical properties of the fluids into which they project. Consider the primary cilia on the epithelial cells that line the lumen of microscopic kidney tubules in which urine formation occurs. The importance of these cilia was revealed when it was discovered that a pair of membrane proteins called polycystin 1 and 2 are localized on the surface of these kidney cilia. Polycystin 1 and 2 form a complex in which polycystin 1 serves as a sensor for fluid flow in the kidney tubules, and polycystin 2 serves as an associated calcium ion channel. Mutations in PKD1 and PKD2, the genes that encode polycystin 1 and 2, lead to polycystic kidney disease, in which the kidney develops multiple cysts that destroy kidney function. Polycystic kidney disease is thought to be a condition that results from a breakdown in the regulation of cell division, because the cysts are the result of an abnormally high level of proliferation of the epithelial cells that line parts of the kidney tubules. According to one model, mutations in the polycystins alter the response of the primary cilia to fluid flow, which leads to a disturbance in calcium flux across the ciliary membrane, which in turn impairs the transmission of signals to the body of the cell and the nucleus, resulting in abnormal cell proliferation. The movement of calcium ions is only one type of signal that is transmitted by the membranes of primary cilia. The primary ciliary membranes of different types of cells contain unique sets of proteins that sense and respond to a variety of extracellular signals. These proteins play an important role in both embryonic development and the maintenance of healthy adult tissues. The importance of cilia in human development is well illustrated by the discovery that Bardet-Biedl syndrome is caused by mutations in any one of a number of genes that affect the assembly of basal bodies and cilia. Persons afflicted with Bardet-Biedl syndrome exhibit a remarkable range of abnormalities, including polydactyly, situs inversus, obesity, kidney disease, heart defects, mental retardation, genital abnormalities, retinal degeneration, loss of hearing and smell, diabetes, and high blood pressure. The fact that all of these dysfunctions can be traced to abnormalities in basal bodies and cilia illustrates the widespread importance of these cellular structures in organ development and function. Many of the genes responsible for these various cilia-based disorders, or ciliopathies, were first identified in model organisms such as Chlamydomonas or C. elegans, which provides another example of the importance of basic research on non-vertebrate organisms in furthering our understanding of human disease. Credits: narrator, name writer, name producer, name editor, name special thanks, names.